I'm going to share my screen and start with the presentation. Okay. Can you see my screen properly?
Yes.
Okay.
Yeah.
You. So today I'm going to go back to some principles in biology that I explained already 3 months ago, but I want to go back because next month Zach and I we're going to mainly Zach is going to present the program and the strategy that we have to find treatment for Myhre syndrome. So you need to really grab some biological principles. I'm not going to transform you in a PhD in science, but just have the basic notion so that you can understand what we do. And since last month, we had some issues with the recording. I'm going to go back and recapitulate a little bit what we said last month about the brain and the nervous system linked to SMAD4. And someone had a question about melatonin, so I expanded a little bit and explained more the link between melatonin and SMAD4. So let's start immediately. Something you need to grab really, the basic principle is the DNA. So what's the DNA? The DNA is when it's packed, is called a chromosome. I think everybody has heard about a chromosome. We have half of our chromosomes coming from our dad and half of the chromosome coming from our mother. So that's the reason why we talk about pairs, chromosome pairs. The DNA, so the chromosomes are located in the cell in a very particular place. It's located in the nucleus. The DNA is made by what we call 4 nucleotides that are called adenine, cytosine, thymine, and guanine. You don't need to remember that, just that it's A, T, and G. So you have, oh, someone has is not mutated. Can you mute yourself? I don't know who's not. iPhone, Lee. If you could mute yourself. Okay, thank you. So the DNA is what we call a double helix. So it's all atoms and molecules that are put together and that forms a double helix. If you look at it, that's a cytosine, that's an adenine, and those pairs stuck one after another makes the DNA. Do you have any here? Does anyone has questions about how the DNA is composed? No? Okay, so I can go further. So basically, I showed you that it's molecule put one after another that makes a long chain. And so when we look at it, we are not, or study it, we are not going to put all the different atoms and molecules one after another. We just put the letters that represents the different nucleotides here. So we write it that way. With it's really those 4 letters one after another. So that's, in fact, that's the beginning of the SMAD4 gene that you see here. So something I want you to understand is that the DNA is located, as I told you, in a part of the cell that is called the nucleus. So if you look here in this figure, that would be the pink ball here, that's the nucleus. And the DNA cannot exit the nucleus. It stays all the time in the nucleus. But the DNA contains the genes that gives the instruction to make proteins. The thing is that the proteins, they are not made in the nucleus, they are made outside of the nucleus, in the rest of the cell. So how can we read the instruction of the gene if it's locked in the nucleus? Well, then we need a messenger to transmit the coded instruction of the gene to go from the nucleus to the cell. And that's what we call a messenger RNA. So it's just for you to understand, why do I explain you that? You can think, well, I don't care about that. I just want to have a treatment for my kid. The thing is that you can hear about treatment that are called ASOs, gene therapy. So you need to understand on what they are acting. An ASO, for example, is going to act on the messenger RNA. That's the reason why I'm explaining you what's a messenger RNA. So you have going back to the basics, you have the DNA and the genes that are in the nucleus. Then the cell makes a copy of the gene, which is called an mRNA. The mRNA has the ability to exit the nucleus and go in the rest of the cells, that is called cytoplasma. You don't need to know that. But then when it's outside, it's going to be, there's the machinery to build the proteins that is going to jump on it and read the code. So the way it's done is that this machinery, which is a ribosome, reads the codes 3 letters by 3 letters. And to each group of 3 letters, there is what we call an amino acid, which is a kind of, think about the Legos, and you have different type of blocks, and you can build a house with different type of blocks, big, small, red, blue, whatever. So those amino acids are the Lego blocks to build the protein. And each small block corresponds to 3 letters. You can see here, this is the code. So you can see that for the same Lego block, the same amino acid, you can have different code. You can have different sequence of 3 letters. So why do I tell you that? It's for you to understand. I imagine you all received the results from the gene sequencing when you had your child or grandchild that had the SMAD4 mutation. I put here the two most common mutation. So you can read on the paper that you have C.1486CT, and then the P. So what does that mean? It means that at the position in the DNA, in the coding sequence, so now you know why it's named coding, because there's the code to put the amino acids to bring the protein. At that location, you had a nucleotide called cytosine that is replaced by thymine, which means that in the protein, you will have an arginine amino acid replaced by a cysteine amino acid. So that's this is the SMAD4 gene, and this is exactly where you have this mutation. So instead of having a C, sorry, I'm admitting Paula or Paul. Instead of having a C in the sequence, you will have a T. So let's go back. Can I go back? Oops, no, that's. So let's see. So you had an arginine that was coded, that is coded usually in SMAD4 by CGT. And now you will have TGT, which is in the codes, the T is replaced by U. So that's UGU instead of CGU. So you have usually a CGU, which is an arginine, and now it's UGU, so you have a cysteine. So do you understand the principle? Do you have any questions? Do you understand now the thing that you read when you received the sequence of your child? No? No questions? Oh, you're smart. you're really smart, all of you. So then if we go to the mutation, the next mutation, which is the isoleucine 500 valine, then you go to a little bit further, and you see that the isoleucine that should be here, so that Lego block that is called an isoleucine, is coded in the SMAD4 gene with ATA. And in fact, the mutation is that instead of this, what's happening here? Okay, sorry, I had messages from Zoom. So I was saying, instead of this isoleucine ATA, you have a mutation with a G here. So instead of having AUA in the code table, you will have a GUA. So going back, oops, go back like that. Let's see, so it's, what did I say? So instead of, where is it? AUA, you will have GUA. Going back, instead of AUA, which is an isoleucine, you will have, what was the thing? GUA. GUA is a valine. So do you have any question? Do you understand what exactly is the origin of the disease of Myhre that you have in your child?
It makes sense to me. I appreciate the explanation.
Okay. So knowing
I'm sorry.
Yeah.
Is it important to know? I understand, but is it important to know the genes or the proteins? Like,
yes.
does that make sense? Like, I mean, I understand the process, but it's hard to keep track of what
So I'm
genes
coming to
are
that.
or the proteins are, and is that necessarily important, or is it just how it's put together that's important?
No, what's important is to know exactly what doesn't work in the person, in the child, in the patient, and what doesn't work in a Myhre patient is the SMAD4 protein.
Yeah.
And
But
now we understand why.
Yeah.
So if you understand why it doesn't work, then you can define a treatment to make it work.
Okay.
So the thing is that we know exactly what's the problem. The problem is a mutation in the gene.
Yeah.
Know exactly where the mutation is in the gene. So what we thought about is the DNA has a mistake in it.
Mm-hmm.
So the best way would be to repair that mistake. If you repair and put this G or T back to this C or A, then you don't have the disease anymore. So how can we do that? There is, in every day, every minute, your DNA is making mistakes for different type of reasons. I listed all the reasons here. For different type of reasons, your DNA make mistakes or is damaged. The reason why there are rare disease and genetic disease is usually a replication error. It means that when you replicate, you multiply by 2 your DNA before having the cells that divides, there is an error. Usually there is a repair mechanisms, the whole machinery in every cell that is going to correct all these errors that happens all the time. With Myhre, the machinery didn't work when it happens. When the error happens, a long time ago, the cells were not able to repair or just missed it. And it happens all the time also. You have repair, and sometimes the repair misses. So the idea that we are going to is we are going to try to correct this mutation. And for that, we are going to use a new technology that is called Remedy. And what's the principle of this technology is that you damage the DNA exactly at the site of the mutation, and then you let all those natural mechanisms repair the DNA by themselves. So you don't put any template, any other DNA in the body, in the patient. You just damage at the right position, and then let nature do its what it has to do. So I'm going to show you how it works. 2643. Wait. 26. Oops.
It's not
43.
something I've heard of, but.
Can you hear the sound or not?
Let's play this real quick so that you have a fair understanding, maybe?
It's kind of low.
Your cells contain chromosomes from each of your parents. These chromosomes carry different versions of the same genes. Sometimes when a person inherits a healthy gene from one parent and a mutated gene from another parent, they develop a genetic disorder. Remedy is a new gene editing approach that can help cells repair mutated genes in cases where the cell contains both a healthy copy and a mutated copy. Remedy uses CRISPR, a gene editing tool, to cut out the mutated gene. CRISPR is very targeted and only cuts the specific sequence the researchers tell it to cut. After CRISPR cuts out the mutated gene, the chromosomes move closer together and begin to communicate. The cell's natural repair machinery steps in and uses the healthy gene as a template to repair the gene that was cut. The result is that the cell will have the original healthy gene on the chromosome from one parent and a corrected gene on the chromosome from the other parent. So far, this has only been studied in the lab on a handful of diseases, but researchers are excited to keep testing this technology to find out if it could someday help patients.
Oops, sorry. Okay, so that's, could you hear properly?
Yes.
Yes.
Oh, okay. So that video is 2 years old. So since then they have, of course, progressed, and they have shown that it works in animal models. So in the mouse it works. So that's the technology we are going to try now. That's one of the things that we are going to try. We try many other things, but that's one of them. Do you understand the principle? Do you have any questions about that treatment?
No, I understood. Doctor, how long this to start this one will be like 3 years? We are still collecting everything, and in 3 years we'll test, or it's already beginning?
No, we are just starting now. So yeah, it will take maybe 3 years before we can go to clinical trials. It doesn't mean that it's gonna be on the market in 3 years. That's impossible. You can do that, but trying in human can be in between 3-5 years. It's very difficult to predict the time frame when you are in drug discovery or in biology, because you may be really lucky.
Yeah, okay.
Everything goes very well, and you can have some issues that you have to address along the road, and it can take a longer time.
Okay, thank you.
Anyone else? Any question? No? I would like to have your reaction to that. Do you like this technology, or do you have any concerns?
Well, my question would be, what does it, if you can go in and repair the cell
Mhm.
to somebody that, to say maybe an adult who has more progressive symptoms of Myhre, what would that look like for somebody? Would it just stop like the over mutation of the gene, and then their body, how would that look for that person? You know, like would all of a sudden their body just start functioning correctly?
Yes, that's the idea
And
about.
would it repair? Would it start to? Would it just like kind of stop regressing at that point, or would it actually get better?
Well, that's a very good
I mean,
question.
yeah, no, I mean, yeah, obviously that's a big question, but
Yeah, so for that we need to try it to really
Yeah,
know. But the
okay.
thing is that well, we need to check, but from the gene editing or the gene therapy we see is that the progression of the disease stops. So you won't have fibrosis anymore, stenosis, but in which extent the damage that were done are going to be corrected by your body by itself, we don't know that.
Yeah.
So, for the autism, for example, we have no idea when the brain suddenly function properly, since you didn't have the formation of the brain that was not correct from the beginning. Can we go back? In theory, yes, it can improve a lot, but it takes a very long time for the brain to recuperate. And I'm going to talk about the brain just after that. But for the fibrosis, for example, for the joint stiffness, then the effect should be in the next weeks or month. That's something that will happen immediately. The only question is for the hearing loss, for everything that is linked to neurology, it takes a long time. And if it's linked to development, it's also the developments have some milestones that you need to reach when you grow up and during the pregnancy. And if you miss those steps, we don't really know yet if you can compensate those steps that were missed or not. Because the gene editing and the gene therapy is quite new. So what we see with the kids with SMA, for example, that have the gene therapy is that they take back all the things that they didn't have before. So suddenly they are able to walk, they are able to open a door when they are 3 years old, which is something that they would have learned before. But thanks to the gene therapy, they can do it. They learn how to do it. For autism, there is not yet. There are some studies. We are not the only one doing that, but it's not yet in humans. So we don't know yet what will be the effect.
Can you answer a question, please?
Sorry?
I have a question.
Yeah, sure. Go ahead.
You have one DNA molecule from your mother and another one from your father.
Yes.
Presumably, in most of these Myhre patients, only one of them has the mutation.
Yes, exactly.
Okay. Okay, if I understand your picture, you strip away, if you take one of those molecules and you strip away the one side of the DNA, but both sides of the DNA have the mutation for SMAD4, correct?
So
How
you
do
cut
you get
it.
information from the mother's DNA to the father's DNA?
Yeah, so the father's DNA, which is usually the father's DNA, has the mutation, so we cut it. We cut the both strands of the DNA.
Well, okay, if you cut both strands, then well, maybe that's the CRISPR protein holds them together. Yeah. I mean, if you cut both strands of a DNA molecule, it can physically separate from the tail end will.
No, not really, in fact, because when you have the whole DNA, it's not you have it's not just the DNA, you have all the proteins, the histones around it, you have a lot of things around it. So it's not that it's going to go apart in the cell. It stays. You just damage one.
Okay.
And
So
it's
the
there.
next question is, how do you get the information from the mother's DNA to fill in for the father's DNA that has now been removed?
So that's a natural process. Is that when the cells comes at a certain point, there is a cell cycle. So that's the cells go through different stages in its life, and there is one of the stages where it was observed that the two chromosomes come close one to another, and that's when you have, in fact, when there is an issue, or even when there is no issue, the two chromosome comes close to another and like exchange their brand of DNA, their strand of DNA, and that's when this phenomenon, if you want, this event can be used to repair the DNA.
I guess I don't understand what you're saying, and I'm sorry for distracting you. I will go research this remedy that you described and see if I can understand it there. Thank you very much.
Well, it's just that if you damage one of the chromosome, the other one comes close. Even if you don't damage, the chromosome come close one to another several times. And when one is damaged, then the other one is used to repair it. That's the natural way to repair the DNA.
I think the disconnect here, Armelle, is that the natural, process that we have to repair DNA is unexpected and amazing that this even occurs in a body, right? It seems unfathomable that the body can do this, and I guess we're just wondering how that works.
Well, we don't know exactly how it works. The first time it was discovered is in yeast. And then it was observed several times in animal cells, like let's say human cells. We don't know exactly how it works, but we know it works. And in fact, the remedy team at the beginning were trying to use gene editing in the other more traditional way, and they were seeing that they have this natural correction, very high percentage of correction, even though they didn't have yet put the template. And at the beginning, they thought it was a mistake. So they repeated the experiment, they repeated, and at the end they said, well, that's not possible that we do so many different times the same mistakes, and other groups of researchers also find these kind of things. So they did more deep experiments, and they realized that in fact it's the natural repair mechanism of the cell that is acting at that level. And so it was discovered at random, as so many different discoveries were done. Do you understand a little bit better, Charles?
Not really. How does this relate? Okay, I'm familiar in E. coli with what's called SOS repair. And that basically works on a single pair of DNA strands.
Mm-hmm. So your issue is that there is more
My issue is how the information in the mother's chromosome gets to the father's chromosome. I just don't understand that. There's no way for DNA to line up. I mean,
oh yes, there is. That's the whole thing. That's every time you have mitosis, you have the chromosome from the two pairs of chromosome that comes and intercale their DNA, and you have swap of the DNA all the time. I can send you some explanation on that, but that happens. That's known already for 35 years that it happens. That's the basic of mitosis. That's the reason why you don't keep always the same exactly chromosome. At the meiosis it happened, and at the mitosis it happens. No, that's something that is known for a very long time. That's not new as remedy is new. That's the reason why you have people that are really different, even in the same family, because it's not that you have exactly the chromosome from the mom and from the father. You have also a remix of the chromosomes. I can send you if you go in the basic books of biology, you will see there are some drawings and figures to explain these kind of things. I will send you that.
Thank you.
Any other questions? No? Then I will
I'm now. Sorry, I lost something because I lost the internet. Why you said is usually is from the DNA father's DNA?
Well, there are some studies that's it's just that they see that it's the case. It's because when I don't like talking about that, because then it's you put the guilt on the father, and we shouldn't do that. There is no guilt, nothing. That's
No,
happened.
of course.
But what's noticed now is that the spermatogenesis, so the way spermatozoids are made, after 30 years old, there is a really the chance to have mutations during spermatogenesis increased a lot in certain genes. So in the genes that are called rasopathy, which is, let's say, after the TGF-beta that I explained 3 months ago, but what we see is that the number of mutation increased terribly after 30 years old.
Okay, thank you.
But yeah.
I understood. Don't worry. Okay, thank you so much.
Okay, any other questions? No. Okay, so I'm going to go to more the recapitulate a little bit what we said last month about SMAD4 and the nervous system. So in the nervous system, just to go really fast, you have nerve cells, of course, neurons. You have the other cells that are called oligodendrocytes, that are the what we call the myelin. So it's around the neuron. So it's like, electrical wire. You have the wire inside, and you have the outside in plastic. So here it's the same thing. You have the neuron with the electrical signal that will go on it, and you have the oligodendrocytes that are on top of it, first to isolate and in fact to accelerate the electricity, the current, to make it go faster, because it's going to jump from, you see that here it's smaller, so it will jump from one node, we call that, to another, and increase terribly the speed of the electrical signal. Other cells are astrocytes, and they are they have a lot of functions. They have a function to ensure the good the capillaries, good functionality of the capillaries in the brain. They are scavengers. They are going to remove all the waste that are produced, because when a cell is working, it makes waste as everything else. So the astrocytes are going to to remove that. They also have a function in stabilizing the synapse. Well, they have a lot of function, and one of the main function is to guide the neurons. So during the embryology, when you form the brain, those astrocytes are going to move and guide the neurons so that the neurons go in the right place. So they are really, in embryology, they are really fundamental. The other type of cell is microglial cells. And do you see it? Yeah. I'm going to move that. Okay. Microglial cells, and those are more the kind of immune system in your brain. So those are the different type of cells. And in fact, all these different type of cells have a link with SMAD4. So there, I took a few publication, research publication here, and where you can find that SMAD4 is involved in neurogenesis and neural outgrowth. So it means that in the formation of neurons and in the way they are going to extend their arms, if you want. SMAD4 is fundamental to have the astrocytes proliferation in adults. So that's really important for us, these kind of things, because it means that if you can correct SMAD4 mutation in the brain, even after you develop the brain, even in adults, you can rectify the astrocytes proliferation in the brain. So that's a good thing for us. So SMAD4 also help the astrocytes to not only to proliferate, but to function properly. SMAD4 is involved in the GABA neurons, which are the inhibitory neurons. So in the brain, you have neurons that are excitatory and neurons that are inhibitory. So that's going to balance. And in fact, that balance between inhibitory and excitatory, when it's not in the proper proportion, is going to trigger autism. So we see that there's also publication about how SMAD4 influence the GABA regulation. The melatonin inhibits the, there's a E, sorry. There's a typo. The melatonin inhibits the SMAD pathway, so it inhibits the end of SMAD4. And the role of TGF-beta has been shown to be to interfere with the peripheral nerve regeneration. So you see that SMAD4 is almost everywhere involved in brain mechanism. The place that is really important for SMAD4 and then for Myhre kids is a part of the brain that is really at the center, that is called the diencephalon. And that's where we see the most SMAD4 expression. So the protein that doesn't work with Myhre, I mean, it's not that it doesn't work, it works too much. The mutation that I showed you before in SMAD4 makes that SMAD4 works too much. And where SMAD4 is a problem when it works too much is that it's here in the center of the brain. Here in the center of the brain, you have hypothalamus that is going to regulate a number of hormones that are going to be released. You have the thalamus that assists the sensory perception, motor function, regulation, consciousness, sleep, and the way you are alert. Then you have another part is the epithalamus that is going to help with the smell. In evolution, we lost a lot of this sense, but at the origin, that was the most important sense. And it also helps with to regulate the sleeps and wake cycles through the pineal glands. And I'm going to explain that further because someone asked that question during the week. And then you have the subthalamus that is involved in movement. Oops, sorry, the other way. there's a part of the hypothalamus that is and the anterior thalamus here. Whoa, it's turning. That is in fact part of the what we call the limbic system, and that's the emotional brain. So that's also relevant for us. So going more in detail, I was talking about the hypothalamus, and the hypothalamus works with the pituitary glands. And the pituitary glands, that's here, there's two parts of the pituitary glands, and it secretes a lot of hormones. And among those hormones, you have the oxytocin hormone that act on the uterus, mammary glands, ovaries. On the other side, you have FSH/LH that works on ovaries and testes. So I'm going maybe too much in details, but basically this pituitary gland is going to work on the puberty and on the development of sexual organ and function of the sexual organs. It acts on the bones tissue, on the skin, on the adrenal cortex of the kidney, which is one of the hormones that makes you alert. That's the when there is a danger that you have also, and it's the stress hormone, and also act on the thyroid. So if we have an issue with the development in Myhre patient of that part, then you imagine that we have an action on all that. So puberty, hormone, bones, skins, and stress, and kidney. So in those, you have an hormone in that part of the brain. You have a hormone that is called anti-Müllerian hormone. And if you look at the function, it's involved in learning and memory, in motor neurons, in bones, in everything that is about puberty and reproduction. And how does that hormone that is produced by that part of the brain that is highly dependent on SMAD4? Oh, did I misplace? Oops, I'm missing a slide. So that hormone, in fact, works through SMAD4. So if you have SMAD4 that works too much, when you have that hormone that comes and try to make its function, it's going to be too much of the action of that hormone. And that hormone is, when it's too much, triggers autism and triggers issues in puberty and polycystic ovary syndrome that we see also with Myhre patient. So that's also somewhere we want to act in a treatment. Do you have any questions? Do I go for you too deep in details? Am I going in the wrong direction? Or are you okay?
I think you're doing great.
It's great, yes. Carry on.
Yeah, it's fine.
Okay, so talking about melatonin. So in this diencephalon that I was talking about, I told you there is the thalamus, the hypothalamus, and just behind at the base of this hypothalamus and thalamus, there's a gland that is the pineal gland. And this gland, that's also the development is very dependent to SMAD4. That gland secretes the melatonin. So I think everybody heard about that hormone. It's a hormone. It's not something. It's not like vitamin B or that. It's really important. It's a hormone with receptors. When the hormone is released, it's going to act through receptors in the cell. And the action of this melatonin is has a lot of different action through the sleep-wake cycle. You know about that. It's neuroprotective. It acts on the hypertension, cardioprotective effects, lipid metabolism, thermoregulation, immunomodulation, the energy, the anti-inflammatory effect and antioxidant effect. So if you have an issue with the pineal glands, went too far. If you have an issue with that part of your brain, then you will have an issue in all those biological effects that I said. The melatonin is not only produced by that gland, it's mainly produced by that gland, but it's also produced by the skin, the bone marrow, the gut, the ovaries, and the prostate. So if you think about Myhre patients, okay, they might have an issue in that part of the brain, but they also have an issue with the skin, the bone marrow, and not really the bone marrow, the end of the bone, sorry, remove that. They have issues with the gut, gastrointestinal issues, and they have hyperfunctioning ovaries or malfunctioning ovaries. So everything, in fact, can be an alert about melatonin. And the receptors, I was telling you, it's an hormone. So it means that it's released in the blood, and then it's going to go through your whole body. And when it hit a cell, is going to go through a receptor through the cell and then have the function inside the cell. So the receptors are fundamental also, because just having the hormone everywhere in your body doesn't mean that it's going to act. You need to have the receptors. And the receptors are expressed, so are present in the central nervous system, cardiovascular system, immune system, skin, liver, kidney, adrenal cortex, so that's where you have the stress, placenta, ovaries, and testes. So again, you can think, well, Myhre patient, they have a little bit of all these issues. Yeah, I went already. I should have put that before. So those are on the right side. You have the different mechanisms of the melatonin, how it works. And one mechanism that is important for us is this mechanism here that is called epithelial mesenchymal transition. So it's a big word, okay, but that's one of the mechanism that is going to trigger fibrosis, which is involved in stenosis. So that's for Myhre patients. Fibrosis and stenosis is very important. And melatonin can block that action. So how does it block that action? You don't need to understand what's here, but it's just to show you that there are many researchers that worked about this EMT, so that process that I just said that's going to trigger fibrosis and stenosis. And what they understood is that the melatonin, in fact, in blocking that action, that process, in fact, is blocking the action of SMAD2/3/4 complex. So the melatonin, the Myhre patients have a SMAD4 that works too much. Okay, so we want to reduce this activity. We don't want to suppress it, because then you trigger other diseases, but we want to just reduce it slowly. If you go to the research, all the research that was published, I just took some examples. Here's a research on melatonin blocking bladder fibrosis. So that's without melatonin, with melatonin, without melatonin, with melatonin, with markers that are typical from this EMT process that triggers the fibrosis. That's bladder, that's renal fibrosis. Same, but they went further. They identified the way melatonin is blocking SMAD pathway. And it's through SMAD, well, you don't need to know that, but melatonin blocks renal fibrosis. Again, in another system, melatonin blocks liver fibrosis through SMAD and EMT. So that's something we really need to take into consideration. And what we can do is to take that structure, that's the molecule, and try to see if we can produce other drugs that are similar to melatonin, or use melatonin in our animal model and cellular model, and see if we can counteract the issues that we see in Myhre patients. So do you have any questions about that? I don't know if we have time. We just have a little bit time. I don't know who was
It's amazing. I'm very surprised with you were talking about Nicole takes melatonin for years because she doesn't sleep, and she has some diverticulum in her bladder, and I'm here thinking, but you said you guys are going to do the study to see how the melatonin can be used for SMAD4 to help more.
That's just an idea. That's just an idea. For now, we have. You'll see next month. Zach is going to explain. We have a lot already on our plate, but we have created this. We are creating the what we call the preclinical platform, where we have all the tools so that we can test drugs in Myhre syndrome patients' cells. And we can add the melatonin to all those, but it's not that we are going to focus only on melatonin. It will be that can be added to the 135 other molecules that we are testing.
Yeah, no, do you guys have some idea how, or it's just an idea and now you guys will study how it can be administrative for helps like now, or it's still beginning? It's beginning yet, you know what I mean?
I can tell you, yeah, take melatonin, that's going to cure your kid. I'm not going to tell you that. The only thing what we can do is take these compounds, put it on the models we have, and see if it works in our model. In theory, studied on other diseases like the liver fibrosis and other fibrosis, we see that at the cellular level, or even in the animal, in mice, it blocks the activity. It reduces the activity of SMAD. Now, if it's going to work in Myhre patients, I don't know. We need first to try to test in the cellular models, in the patient's cellular models that we have, then in the mouse model that we have, and then I can tell you, well, there is a big chance that it will work, but still, that the only way to be sure is to go then to human. So it's a long process, and the theory and the data, the research from other diseases says or points towards the direction that it could work for Myhre.